Here's something that will probably make you look at your hands differently after today. Hold out both hands, palms down. Look at your index finger and your ring finger. Are they roughly the same length, or is one noticeably longer than the other? That little detail, something most people have never thought about in their entire lives, is actually a biological record of what your hormones were doing while you were still in the womb. We'll come back to that, but first, I want to ask you something even more fundamental. Do you actually understand what a hormone is? Not the word, not the general vibe of it makes you feel things, the actual mechanism. Because the second you understand how hormones genuinely operate, you'll realize that nearly everything you've been told about them is incomplete, and a couple of things you believe are just flat out wrong, including, by the way, the story about which hormone is responsible for masculinizing the brain. That one's going to surprise you. Let's build the foundation first. A hormone is not the same thing as a neurotransmitter. Neurotransmitters work locally. They jump across a tiny gap between neurons and affect what's right next door. Hormones are something else entirely. They're produced in one place, usually a gland, like the thyroid, or the testes, or the ovaries, or the adrenal glands, and then they travel through the bloodstream to exert effects somewhere else entirely. That's the key feature. Distance. One location produces it, another location responds to it. And hormones can work fast, like adrenaline, which can spike your heart rate in seconds, or they can work slowly and profoundly, like the sex steroid hormones, estrogen and testosterone, which are what biologists call lipophilic. They're fat-loving molecules that can actually penetrate cell membranes and get into the nucleus of a cell, where they directly interact with DNA and change which genes get expressed. That's not a fast reaction. That's a rewrite, and that distinction matters enormously when we're talking about development. Because here's the thing, we all went through a biological process that most people don't know the details of. Starting before birth, a cascade of hormonal signals determine the structure of your body, your brain, your spinal cord. And that process is way more layered than girl chromosomes make a girl, boy chromosomes make a boy. There are actually 4 distinct steps, and they don't always line up the way you'd expect. Step 1 is chromosomal sex. This is the XX or XY distinction most of us learned in school. 2 X chromosomes, or 1 X and 1 Y. That's the starting blueprint. Step 2 is gonadal sex, whether the developing fetus forms ovaries or testes. In a typical XY development, genes on the Y chromosome, including one called the SRY gene, promote the formation of testes, and crucially, the Y chromosome also carries the instructions for something called Mullerian-inhibiting hormone, which actively suppresses the development of the female reproductive structures. So it's not just building one thing, it's simultaneously shutting down another. Step 3 is hormonal sex, the effects of testosterone, estrogen, and their derivatives on how the body actually takes shape. And step 4 is morphological sex, what you can actually observe, body structure, external anatomy, everything visible. 4 steps, and they don't always produce the same result. Here's one of the most striking examples in all of biology, and it comes from a real community in the Dominican Republic. Some individuals are born with a rare genetic mutation. The enzyme that converts testosterone into a more potent androgen called dihydrotestosterone, or DHT, doesn't function. That enzyme is called 5-alpha reductase. And because DHT is what drives the development of external male anatomy in utero, these babies are born without the typical male external anatomy. Even though they have XY chromosomes and internal testes, they're often raised as girls. Then, somewhere around age 11 or 12, puberty triggers a surge of testosterone from those internal testes, and because the secondary development doesn't depend on DHT the same way, the external anatomy begins to develop in a more typical male pattern. These individuals have a name in their community, Guevedoces, which translates roughly to penis at 12. It's one of the most dramatic illustrations in biology that chromosomal sex, gonadal sex, and external anatomy can tell 3 entirely different stories at once. And cultural outcomes vary enormously. Some choose to identify and present as male after this transition, others don't. And some communities have developed a broader third category to accommodate this experience. Now let's get to the twist that I promised you. You might reasonably assume that testosterone is what shapes the brain in a more masculine direction. After all, it masculinizes the body, right? Wrong. Here's what actually happens. Testosterone can be converted into estrogen by an enzyme called aromatase. And it is that converted estrogen, not testosterone, not DHT, that organizes the circuits of what we call the masculine brain. You heard that correctly. Estrogen masculinizes the male brain. It sets up the neural architecture, and then later, testosterone activates the behaviors those circuits support. One way to see this principle in action is in a condition called androgen insensitivity syndrome. These are individuals with XY chromosomes and functional testes, but their cells lack working receptors for testosterone. So even though testosterone is being produced, the body can't respond to it. What you get is an external appearance that is entirely female, because without testosterone binding to its receptors, the body develops along a default pathway. They report feeling and identifying as women. Their chromosomal sex says XY. Their gonadal sex is technically male. Their hormonal sex overrode everything downstream. It's a remarkable natural demonstration of just how much receptor function matters. A hormone that can't reach its target is physiologically invisible. You can actually see the effects of DHT written right on the outside of people's faces and heads, though most people don't know how to read the signs. DHT drives facial hair growth by binding to receptors in the face. It also promotes hair thinning on the scalp by binding to receptors there. Different locations, opposite effects, same molecule. The drugs used to treat hair loss, known as 5-alpha reductase inhibitors, work by blocking the conversion of testosterone to DHT, which is why they can reduce hair thinning but also carry significant side effects, since DHT is involved in libido, connective tissue repair, and drive. There's also a genetic component. Whether your hair thins and where depends on the distribution of DHT receptors on your scalp. Your mother's father is the traditional predictor, since the relevant receptor patterns are often inherited through the maternal line. Creatine, one of the most popular and well-researched sports supplements in the US, appears to increase 5-alpha reductase activity, which means it may increase DHT levels. For some people, that translates to faster hair thinning. For others, it doesn't seem to matter. The effect varies by individual, but it's worth knowing, especially if you're predisposed. Now let's talk about something that should genuinely concern everyone listening, especially if you're in a phase of life involving pregnancy, puberty, or planning a family. Sperm counts in the United States and other industrialized nations have declined by roughly 50% over the last half-century. That's not a blip. A major meta-analysis found an approximately 50% drop in sperm concentration in males from industrialized regions between 1973 and 2011, and more recent analyses confirm that declining trends have continued into the 21st century. The World Health Organization now lists infertility as a public health priority, with male factors accounting for more than half of all cases. What's driving this? A significant part of the answer points to endocrine disrupting chemicals, substances in our environment that interfere with how hormones function. One of the most well-documented is a herbicide called atrazine, widely present in waterways across the United States and studied extensively by researchers at UC Berkeley. In areas with higher atrazine concentrations, male frogs showed testicular malformations at rates between 10 and 92%. This isn't fringe research. It's peer-reviewed, federally funded, and replicated. Atrazine doesn't just show up in the frog pond, it's in a lot of the water supply. Another concern that often flies under the radar: evening primrose oil, which is common in skincare products. It has significant estrogenic chemical properties, and there have been documented cases of boys developing premature breast tissue through skin contact with products containing it. Whether you're trying to tip your hormones in a more estrogenic or more androgenic direction, awareness of what's in your environment matters. Skin absorption is real, and children are significantly more vulnerable to these effects during developmental windows than adults are. Cannabis also deserves mention here, not as a moral or legal judgment. Those conversations belong elsewhere, but as a hormonal one. Multiple studies point to the fact that components of the cannabis plant promote aromatase activity, which means they accelerate the conversion of testosterone into estrogen. Increased exposure to endocrine-disrupting chemicals beginning in prenatal life may represent a significant contributing factor to the temporal decline in sperm counts observed in recent decades. Alcohol, particularly grain-based options, can also increase estrogenic activity in the body, which is why its use during puberty and pregnancy is a concern beyond the more widely known effects. Puberty doesn't have a clean start and end date. It's a gradual biological transition, and hormonal exposure during that window shapes development in lasting ways. Cell phone radiation is a newer variable in this conversation, and honestly, the research is still developing. But there's a growing body of animal and human studies suggesting that prolonged proximity of phones to the reproductive organs may affect hormone profiles, including testosterone, thyroid hormones, and in young women, prolactin. One study in clinical biochemistry found significant hormonal differences in people based on how much time their phones spent near their body and how close they lived to cell tower base stations. Is that enough to draw firm conclusions? Not quite. Is it enough to make you think twice about where you keep your phone? Probably yes. Now let's come back to your fingers. The ratio of your index finger to your ring finger, what researchers call the 2D to 4D ratio, turns out to be a measurable marker of how much androgen you were exposed to while you were developing in the womb. In people with higher prenatal androgen exposure, the ring finger tends to be noticeably longer than the index finger. In people with lower prenatal androgen exposure, the two fingers tend to be more similar in length. This ratio is present at birth and doesn't change, which is what makes it such a useful research tool. It was published in Nature in 2000 and has been replicated multiple times. The key point isn't that you can read someone's full hormonal biography from their hand. You can't. The data are averages, not predictions for individuals. But it does demonstrate something profound. The hormonal environment you were in before you were even born left a permanent, measurable signature on your physical structure. And researchers have found similar fingerprints in studies on auditory differences and neural architecture, suggesting that prenatal androgen exposure may correlate with aspects of brain organization, including, in some studies, sexual preference. This is active, ongoing science, not settled doctrine. But it underscores a core principle. Biology is shaping us in ways that begin long before we're aware of ourselves. One of the stranger examples of this whole system at work, and it really is stranger than anything a screenwriter would come up with, is the spotted hyena. Female spotted hyenas are dominant in their social structure, and they have external genitalia that is anatomically larger than that of the males. This is driven by extraordinarily high levels of a precursor hormone called androstenedione in the females. These animals are a living demonstration that androgenization of peripheral anatomy is not exclusive to one chromosomal type. It's driven by hormone levels and receptor distribution, full stop. Biology doesn't follow the simplified textbook story. It follows chemistry. So here's the practical takeaway. You are not just who you are because of a single chromosome or a single hormone. You are the product of a long, layered developmental conversation between genes, hormones, enzymes, receptors, and environment, starting before you were born and continuing right now. Knowing this changes how you relate to your own body, and it changes how you relate to the biological complexity of everyone around you. The tools here are awareness. Check what's in your skincare products. Think about where your phone lives during the day. Pay attention to alcohol and cannabis use, especially during developmental periods. And if you're planning a family, know that the hormonal environment during pregnancy matters more than most people realize. Before our next episode, here's your challenge. Look up your 2D to 4D ratio. Measure carefully from the base crease to the tip on your right hand, and then drop it in the comments with a simple number. You'll be contributing to what is genuinely one of the most interesting informal data collections on this platform. I want to see what this community looks like, and I think you'll be surprised by the range. Growth is not accidental, it's intentional. If you're ready to build a better version of yourself, welcome. Stay tuned.